In addition to sperm, seminal fluid contains multiple signalling factors that promote a male's reproductive success, sometimes against the best interests of his partner. In male Drosophila, for example, reproductive accessory glands secrete factors into the seminal fluid that alter the behavior of recipient females so that they are less inclined to remate with other male flies. Clive from the University of Oxford was interested in how the accessory gland produces these secreted factors.
We'd started to work on the cell biology of the system, how were these secretions being made? And a student in the lab, Corrigan, who's the first author on the paper, discovered that there seemed to be vesicular-like structures within the lumen of the gland.
These vesicle-like structures could represent exosomes, small membrane-bound vesicles that form inside late endosomal multivesicular bodies and are then released at the cell's plasma membrane.
Exosomes, in principle, carry not only ligands that will bind to target cells, but they'll also carry receptors and intracellular signaling molecules inside them. So they potentially have a lot of possibilities in terms of their ability to reprogram cells. And this idea that males will have different interests compared to females, the concept of sexual conflict. One way of a male persuading a female to behave in a particular way is not only to transfer to her ligands, but also receptors and downstream signaling molecules that would overpower the signaling cascades within the female cell itself.
Working in collaboration with Deborah Goberdhan's laboratory, Corrigan et al. Expressed the human exosome marker CD63 in the different cell types that make up the accessory gland. CD63 was only packaged into vesicular structures and secreted into the accessory gland lumen when it was expressed in the gland's so-called secondary cells. The researchers therefore took a closer look at CD63's intracellular localization.
It's trafficked on membranes within a number of intracellular compartments, but we can see distinct and very small puncta within a specific acidic domain within each cell. And using different types of markers, we were able to show that that particular structure seemed to be a multivesicular body.
The multivesicular bodies and endolysosomal compartments of secondary cells are exceptionally large, allowing the researchers to follow the dynamic interactions between these organelles and even observe the formation of vesicles in the interior of the multivesicular bodies, a process that isn't readily visible in other cell types.
For example, we were able to see invagination of membrane, which carried the CD63 marker inside the multivesicular body, and occasionally then its resolution into what appeared to be a puncta within the compartment, which we then assumed was a newly formed exosome.
Indeed, the number of exosomes secreted into the accessory gland lumen was drastically reduced when the researchers knocked down proteins required to generate the vesicles inside multivesicular bodies. As a result, smaller amounts of CD63 were transferred to female flies during mating. But what happens to the exosomes next?
When the male mates, these fluorescent exosomes will fuse to a number of different structures within the female. First of all, they fuse with the membrane of the sperm that's been transferred together with the rest of the seminal fluid. We can visualize that by the change in the shape of the GFP as it actually fuses to the plasma membrane of the sperm. And then it also seems to interact with some of the female epithelial cells, so we can see the exosomes lining up along the surface of the epithelium. We've no evidence that those exosomes then get internalized in any way at the moment, but they certainly sit on the epithelium, and so potentially they could be signaling from that site as well.
Exosomes secreted from the prostate gland can fuse with human sperm in vitro, potentially promoting the sperm's full activation. Whether fly exosomes have a similar role is unclear, but Corrigan et al. Focused on the vesicle's potential effects on female physiology and behavior.
So we already knew that the secondary cells can have very specific roles on the behavior of females after mating, so that when we reduce BMP signaling within those cells, females no longer have the full post-mating response where they prevent other males from mating with them. And when we reduce the amount of exosomes that were being produced by secondary cells, the females were not fully able to reject males after they'd mated with the exosomeless male. And this suggested to us that somehow the exosomes are involved in this specific behavioral reprogramming that the secondary cells are involved in.
This result also suggested that BMP signaling might somehow regulate the production of exosomes by accessory gland secondary cells.
When we reduced the amount of BMP signaling within the secondary cell, vesicles labeled with CD63 accumulated within an acidic compartment within the cell, but then those vesicles were not able to actually be trafficked out of the cell. And that led us to postulate that the BMP signaling pathway was in some way controlling endolysosomal trafficking. And then when we look back at cells where BMP signaling had been hyperactivated, what we were able to see was that we were able to generate a huge lysosomal structure within those cells. And that sort of supported our idea that one of the key events that BMP signaling is controlling is the trafficking of membrane and membrane proteins into late endosomal compartments and into the lysosome, as well as to the surface of the cell to release exosomes.
Corrigan et al. Are now interested in whether BMP signaling regulates exosome secretion in human tissues such as the prostate gland, and, explains Deborah Goberdhan, whether this process contributes to tumorigenesis, since prostate cancer cells are known to inappropriately secrete exosomes into the bloodstream.
Cancer cells are known to release large numbers of exosomes, and these have been implicated in reprogramming other cells and priming the pre-metastatic niche. So the data from the fly has suggested that normal secondary cell exosomes have the ability to mediate events associated with sexual conflict, such as the physiological reprogramming of females. And one might imagine that prostosomes, the exosomes secreted by prostate cells, may also have functions in reprogramming other cells. When cancer cells lose their polarity and secrete more exosomes into circulation. So what we're trying to do with work in cell culture is to try and isolate exosomes from cancer cells, look at the cargoes, the content of those exosomes, and then try to look at the effects of those different isolates on cell migration or angiogenesis or facilitating metastasis.
For now, though, you can learn more about how BMP-regulated exosomes from Drosophila male reproductive glands reprogram female behaviour in the paper by Corrigan et al., published in the September 1st, 2014 issue of the Journal of Cell Biology.
You